Sir, namaste. Harish sir.
ನಮಸ್ತೆ.
Everything is set, sir. Almost done.
ಓಕೆ, ಇದು ಅವ್ರು ಅತೀರಾ ಅವರು ನಮ್ಮ ನೊಜೊಮಿ ಸೆನ್ಸನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾರಲ್ಲ, ಜಸ್ಟ್ ಒಂದು 2 ಲೈನ್ ಅಲ್ಲಿ.
ಓಕೆ ಸರ್. ಎಸ್ 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 ಸರ್.
ಓಕೆ, ಹಾಗೆ ಮಾಡಿ ಆಯ್ತಾ?
ಹೌದು, ಫಸ್ಟಿಗೆ ಇಂಟ್ರೊಡಕ್ಷನ್ ಇರುತ್ತೆ ಸರ್, ನಾನೇ ಮಾಡ್ತೇನೆ. ದೆನ್ ಆಫ್ಟರ್ ದಟ್ ವಿಲ್ ಕಂಟಿನ್ಯೂ.
ಓಕೆ, ಯಾ.
ಸರ್, ವಿ ಆರ್ ಆಲ್ ಸೆಟ್.
ಓಕೆ, 1 minute ನಾನು ಕೇಳ್ತೇನೆ. ಐ ಥಿಂಕ್ ಆಲ್ಮೋಸ್ಟ್ ಡನ್. ದೇ ಆರ್ ಲಾಗಿಂಗ್ ಇನ್.
ಓಕೆ, ಎಸ್ ಸರ್.
ಯಾ.
Minna-sama, konnichiwa. Honjitsu no shikai wo tsumemasu. Atira desu. Yoroshiku onegaishimasu. A very good morning and namaste to one and all present here. I am Atira and I will be your host for the event. 28th February is celebrated as National Science Day to commemorate the discovery of the Raman effect by Nobel laureate and physicist Sir Chandrasekhara Venkata Raman. On this special occasion, I welcome you all to the Sakura Science Online Exchange Program, an Indo-Japan Bination seminar on biomedicine and biotechnology organized in conjunction with the Faculty of Medicine, University of Miyazaki, Japan. The seminar will primarily focus on biophysical techniques for biomedical research and skill improvement. I am extremely elated to be able to welcome today's panel of esteemed educators. I request Dr. Venkatesh Kamath, Assistant Professor, Grade 3, Department of Biotechnology Engineering, and Deputy Controller of Examinations, NMAMIT, to give a brief introduction about the program and the panelists. Over to you, sir.
Namaste and good morning to everyone. Myself, Dr. Venkatesh Kamath, Organizing Secretary of this SSP event. It is my pleasure to welcome all the esteemed guests and the participants. The Sakura Science Online Student Exchange Program with the theme Indo-Japan Bination Seminar on Biomedicine and Biotechnology, organized by the Faculty of Medicine, University of Miyazaki, Japan, in association with Department of Biotechnology, N.M.A.M Institute of Technology, Nitte, India. The topic of this program focuses on biophysical techniques for medical research skill improvements. In this program, we have 5 invited lectures, an ideathon student paper presentation, and a virtual lab tour to Central Instrumentation Facility at University of Miyazaki, Japan. I welcome Professor Dr. Nozomi Watanabe, Chair and Head, Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki, Japan, and also the Seminar Chair of this event. Welcome, Madam. We have with us Dr. Niranjan and Chiplunkar, Principal, NMAMIT Nitte. Welcome you, sir. I welcome the Vice Principals, Dr. Dr. I.R. Mittantaya and Control of Examination, Dr. Srinivas Rao, for the event. I welcome the convener of this seminar, Dr. Ujjwal P., Professor and Head, Department of Biotechnology, N.M.A.M Institute of Technology. Welcome you, Madam. I welcome Dr. Harish Kumar Madhyastha, Associate Professor, Faculty of Medicine at University of Miyazaki, who is the convener of this seminar and also the coordinator of the SSP Online Exchange Program. I take this opportunity to welcome our guest speakers of the event, Dr. Kaoru Ohe, Dr. Betasivi Urbasari, Dr. Radha Madhyastha, and Dr. Yuichi Nakajima from University of Miyazaki, Japan. And I also welcome Dr. Praveen Kumar Shetty, Director R&D, Nitte deemed to be University, and Dr. Shyam Prasad Sajankila of N.M.A.M Institute of Technology, Nitte, for this seminar. I welcome all the invited guests and invitees from the Faculty of Medicine, Faculty of Engineering of University of Miyazaki, Japan, Department of Biotechnology, and the N.M.A.M Institute of Technology, Nitte, and Nitte deemed to be University for this program. I welcome all the student participants who are attending from Shambhavi Seminar Hall as offline attendees, which is where this program is going on in a dual mode, that is online and offline mode. I welcome all the participants to this program. I welcome one and all everyone for this event. Hope we have a fruitful interaction over the day. Over to Athira.
Thank you, sir. I take this opportunity to welcome Dr. Nozomi Watanabe, Chair, Professor and Head, Department of Cardiovascular Physiology, Faculty of Medicine, and Deputy Director, Heart Center, University of Miyazaki Hospital, Japan. A warm welcome to you, ma'am. I request Dr. Nozomi Watanabe to deliver the inaugural address.
Hello, everybody. I'm Professor Nozomi Watanabe. On behalf of the Division of Cardiovascular Physiology, the University of Miyazaki Faculty of Medicine, it is a real pleasure and a privilege for me to participate in the opening ceremony of the Sakura Science Online Exchange Program, Indo-Japan Seminar of Biomedicine and Biotechnology, organized by the Nitte University and the University of Miyazaki. As the chair of this exchange program in science, I am proud to have been given the responsibility on this important international program, especially in this difficult time of COVID-19 pandemic. It is with the idea of encouraging and accelerating the exchange of information and opinions on biomedicine and biotechnology that this program was organized. It is certainly a pity that we cannot see each other face-to-face in this wonderful exchange program. But it should be great to be able to share this one-day seminar with distinguished colleagues in India, even online. I hope we will be able to have a fruitful discussion today, and we look forward to our continuous collaboration between our universities. Thank you. Have a good day.
Thank you for your wonderful words, ma'am. May I now request the Principal of N.M.A.M Institute of Technology, Nitte, Dr. Niranjan Chiplunkar, to share a few words and deliver the opening remarks.
Hello, everyone. Good morning to all my dear participants in India, and good afternoon to all of you from Japan. Dear Madam, Professor Dr. Nozomi Watanabe, Dr. Madhyastha, all my colleagues here at Nitte University, and also the resource persons from Miyazaki University, and all my dear students who are participating on this program through online. It gives me an immense pleasure to be part of this inaugural program of online Sakura Science Exchange Program with the theme of Biomedicine and Biotechnology, that is Biophysical Techniques for Biomedical Research Skill Improvement. We are from the engineering faculty of Nitte University, and I'm aware that Miyazaki University has been having very fruitful collaborations and deliberations for past several years with Nitte University's medical team, and it is the first time that engineering faculty is being part of this particular program. And I wish and hope that in the days to come, in the years to come, we will have more such interactions, not only online, even offline interaction with University of Miyazaki. Few years back, I had the privilege to visit Miyazaki University, and along with our Pro Vice Chancellor, Professor Muritaya, and that time Dr. Harish Madhyastha helped us and took us around the university facilities, and then it was really we were very happy to see various facilities and infrastructure and the various research facilities at Miyazaki University. We being from engineering fraternity, we always believe that whatever engineering and technology developments that happen finally must be of some use to the other profession, and the medical profession, and then medicine and biotechnology profession is one important application area where engineering technology can really come in a big way. In the past few months, our engineering faculty, few years in fact, we have been working very closely with our School of Medicine, School of Dentistry and Pharmacy of Nitte University. But I believe this will be our first opportunity to even interact with the medical related faculty from a university abroad. So we are really very thrilled for this. Our institution has been working with another Japanese university, that's Ritsumeikan University, for Sakura exchange program for last 5 years. Only thing is, for last 2 years, actually the exchange of students could not happen because of this corona pandemic. But earlier to that, our students used to go to that university, as well as students from Ritsumeikan used to come here. I believe if once the corona situation, settles, becomes everything fine, then we would like to even invite students from Miyazaki University to our university. And there's a lot of possibilities wherein both of us can work together. And I once again take this opportunity to thank Madam Professor Dr. Nozomi Watanabe for being there, and also the entire Miyazaki University team for giving us an opportunity to be associated with you in this very important program of Sakura Exchange. I earnestly appeal to my student friends who are watching this program online to be interactive with all the resource persons, and see that you get the maximum out of this particular day-long program. Wishing the program every success. Thank you very much. Thanks for the opportunity.
Thank you, sir. I now invite Dr. Ujjwal P., Professor and Head, Department of Biotechnology Engineering, N.M.A.M Institute of Technology, to deliver a note on intra-departmental activities conducted here at NMAMIT. Over to you, ma'am.
Good morning from India and good afternoon to Japan. Ladies and gentlemen, respected members of Miyazaki, Japan, my principal, vice principal, my colleagues, and dear students. To have a gist of the department, Bachelor of Biotechnology Engineering program started in 2002 with an intake capacity of 60 to train students in biological applications with engineering principles. We are autonomous, attained under VTO Belagavi from 2007 onwards. VJST, Center of Excellence, we are involved in a number of interdisciplinary projects. We have student exchange program with foreign universities. We are continuously involved with students and faculty research programs, wherein we have a total grant received amounts up to date till 2.5 crores, with many publications in reputed journals. We have experienced faculties in the field of engineering and biological sciences, and all the faculties have completed their PhD programs. Our faculties are also guiding external PhD candidates. Our faculties are involved in various themes of research, which includes nanosciences, biofuels, bioremediation, microbiology, biopharmaceuticals, etc. Our laboratories are well equipped with various equipment. Students and faculties are continuously involved in competing with day-to-day challenges in terms of research and development, and have received awards with different authorities. Our department, NMAMIT, is one among the 32 information and demonstration centers by Karnataka State Bioenergy Developmental Board with an annual grant of 4 lakhs, wherein we are producing biodiesel from various non-edible oil seeds as well as from used cooking oil from the district around. This particular program creates awareness, disseminates information about biofuels among the scientific community, students, farmers, public, and demonstrates the technical know-how of the biodiesel production. The center was established in the year 2011. This seminar today is expected to display significant developments in the important areas of biophysical techniques for biomedical research and their relevance to the welfare of mankind. It's my pleasure welcoming all the participants, faculty researchers, students for this seminar 22. And I hope this seminar will enhance everyone's knowledge on novel concepts and thereby stimulate our minds to dwell into further research. I wish all the students and participants every success and look forward for the outcome. Thank you and all good wishes from biotech family of NMAMIT Nitte.
Thank you for your words, ma'am. The first talk for the session is on pollution in Japan and development of high-performance arsenic adsorbents by Dr. Kaoru Ohe, Department of Applied Chemistry, University of Miyazaki, Japan. I call upon Ms. Anjali Rao to give us a brief introduction about Dr. Kaoru Ohe. Over to you, Anjali.
Dr. Kaoru Ohe is a faculty of engineering at the University of Miyazaki. Dr. Ohe began her academic career in 1999 as an assistant professor at the Department of Applied Chemistry, University of Miyazaki. She was chosen as a research scientist for the bilateral program supported by JSPS and School of Chemistry, the University of Melbourne, between September to October 2009. In 2014, Dr. Ohe completed her PhD in engineering at the Interdisciplinary Graduate School of Agriculture and Engineering, University of Miyazaki. She became the Associate Professor at the Center for Science and Engineering Education, Faculty of Engineering, University of Miyazaki in 2019. She is currently working on the development of adsorbents for the removal of hazardous materials in aqueous solutions. Dr. Ohe has a total of 57 publications to her credit and has proceedings in 73 conferences. It's a great pleasure to have you here with us, ma'am. Over to you.
Hello. Hello.
Yes, sir.
I think we are what you call ahead of time. Now it is we are 25 minutes ahead. I think OH is not yet logged in.
Okay, sir.
If chair approves, Professor Kamath approves, we can have the next speaker now. By the time I will inform OH to be in the line.
Alternatively, sir, you can engage with the SSP plans, which you would have
Okay.
otherwise.
Okay, okay, okay. So Professor Ohe will join soon because it is 20 minutes earlier, so that I am just introducing the activity of SSP. SSP, Sakura Science Program. Am I audible?
Yes, sir.
Okay, Sakura Science Program, which is funded by the government of Japan, under the agency of the JST, Japan Science and Technology wing. It is actually to encourage the young faculties and students of Asian countries, including India. We have been conducting this program from 2016 onwards till now, till 2019, we have done virtually invited more than 180 members from India and total 52 members from the Nitte University. Of course, as Professor Chiplonka rightly mentioned, engineering college, of course, it is the first time we are organizing the joint event. Otherwise, Nitte University, Derlakata campus, medical college, dentist, pharmacy, and biomedical science, they are the most beneficiaries. And of course, Dr. Venkatesh Kamath visited here. And from that onwards, day onwards, we literally engaged with joint activity with Engineering College of Nitte Trust of Nitte University. Basically, we invite ordinary these faculties and young students for 1 week or 10 days, which is completely funded by all expenses will be borne by the government of Japan. We expose, we give the training to the different high-end instruments, which you will be seen in the last wing of this today's session, the last wing. We will give the training and hands-on training and various biological experiments which are being conducted in our routine research activity in our lab and some other labs of medical college and agriculture faculties and engineering faculties also. In between, we also give a sort of social impact of the how Japan society is nurtured, cured, and what is their day by day living style. We also once or twice we conducted a homestay in the Japan village, and we stayed overnight over there when we actively participants of the Indian universities actively participated in their day-to-day life with agriculture activities, fishing activities, and other what you call Atmanirbharata style. So in that way, this is a what you call a small stepping stone for the further intra-departmental or intra-university activities. So to link further, wherein continuous project and continuous activity and sustainable activity in the areas of the human development and human-to-human communication and good living and with good health. This is the ultimate theme of this entire program, which have been conducted in India, particularly under SSP program till now. Fine. Because of this activity, several Indian students already visited us as a SAP, that means Semester Abroad Project for the dissertation program, and they stayed here for 6 months and conducted their dissertation work in our lab. Several students came. And now a joint project is going on with Nitte University faculty, Kshema, the Faculty of Medicine, where total of 1.1 crore rupees joint activities going on. This is the second year is going to over in this March. From the April, the third year is going to be then. The theme of that project is the child development under the hotspot environmental areas of the Mangalore, Kasaragod, Udupi, as well as Hassan district of Karnataka. And as you all know that some 1980s, in the early 80s and late 70s, there was a huge problem for endosulfan, where the cashew nut plantation where active was there in, still it is there, and the endosulfan spray, aerial spray made a havoc of the entire population. And till now, some pockets of this Belthangadi, Mangalore, Kasaragod, and some other areas are still suffering from kind of some genetic abnormalities, and some stillbirth they are observed. When our president, ex-president Professor Ikonome visited Mangalore University for the convocation program of Nitte University some 3-4 years before, that time he has picked up these issues and asked us to draft a joint project with JSPS. We drafted the project and project got clicked. And this is how the genesis of SSP, Sakura Science Program, ultimately converts into the research activity, joint research activity with other institute, with Miyazaki, University of Miyazaki, and finally it culminates into a sustainable activity of with in terms of the projects. That's how our our theme, university slogan conducts. University slogan says, "Look at the world, start with the community." So, as Professor Chiplunkal rightly mentioned, once again I'm telling, this is the first time we are having the online version of this Sakura Science Program, and certainly there are joint activity or joint platforms which can be further propagated or multiplied with the engineering college, so that a transdisciplinary activity or interdisciplinary activity with the biology as well as the technology can be clubbed together for the well-being of the animals, humans, and plants. Thank you, sir.
Sir, whether madam is logged in?
1 minute, just I think she has. No, not yet. Anyway, we can ask the next speaker to be in the Dr. Betasavi to present her this one. By the time I will call Dr. Ohe to be this one. Is it okay?
Fine, sir. But it would take about half an hour, right, sir? Plus scheduling. That's fine.
How many?
We have allocated around 40 minutes. We have allocated, sir.
Okay, no problem. She's prepared for the 40-45 minutes. No problem.
Okay. Yeah.
Now,
Atira, please, yes, sir. We'll introduce madam. Then we'll start.
Yeah.
Atira, please do the needful.
Yes, sir. The first session for today is on the topic Ways to Study Cellular Inflammation by Dr. Betasavi Purbasari, Faculty of Medicine, Brawijaya University, Indonesia. I call upon Ms. Rashmita to briefly introduce our guest speaker. Over to you, Rashmita.
Dr. Beta is a neurologist, earned her MD from Brawijaya University, Indonesia, and PhD from University of Miyazaki, Japan in the year 2022. Her recent research focuses on the role of cholinergic agonist in macrophage inflammasome modulation. She intends to be a physician scientist that can integrate biomolecular research and medical science to be used in the clinical application for patient care, especially in neurology field. She is currently attached with Bravijaya University Faculty of Medicine as faculty. Over to you, ma'am.
Good afternoon, everyone. Can everyone see my screen now?
No, ma'am, it's not yet visible.
Is it shared now? Still not yet shared?
Not yet.
Madam, possibly at the right bottom, there is a pop-up you may be getting. Dismiss option is there, blue colored pop-up, right bottom. It may be prompting you to dismiss that notification.
Dr. Coher has joined. Kauro joined, already joined. Can we reshift it?
Yes, sir. No, sure, sir.
Yeah, she already joined.
I'm sorry. I'm late.
No, you are early actually, no problem. ORSNC Hiroshi Konogaishima, Chotamate. Atira, can you please introduce Dr. Kaoru Ohe once
Yes,
again?
sir. Yes,
Yeah.
sir.
She's already joined.
The first talk on the session is on pollution in Japan and development of high-performance arsenic adsorbents by Dr. Kaoru Ohe, Department of Applied Chemistry, University of Miyazaki, Japan. I call upon Ms. Anjali Rao to give us a brief introduction about Dr. Kaoru Ohe. Over to you, Anjali.
Dr. Kaoru Ohe is a faculty of engineering at the University of Miyazaki. Dr. Ohe began her academic career in 1999 as an assistant professor at the Department of Applied Chemistry, University of Miyazaki. She was chosen as a research scientist for the bilateral program supported by JSPS and School of Chemistry, the University of Melbourne, between September to October 2009. In 2014, Dr. Ohe completed her PhD in engineering at the Interdisciplinary Graduate School of Agriculture and Engineering, University of Miyazaki. She became the Associate Professor of the Center of Science and Engineering Education, Faculty of Engineering, University of Miyazaki in 2019. She is currently working on the development of adsorbents for the removal of hazardous materials in aqueous solution. Dr. Ohe has a total of 57 publications to her credit and has proceedings in 73 conferences. It's a great pleasure to have you here with us, ma'am. Over to you.
Thank you for invite me and me. And so good morning, everyone. Nice to meet you. I'm Kaoru Oe from the Faculty of Engineering at Miyazaki University. I would like to thank you for giving me this opportunity to talk at the Sakura Science Program between Nitte University and the University of Miyazaki. Today, I would like to give you presentation on pollution in Japan and development of high performance arsenic adsorbents. So I will share the slide.
Yeah, slide is visible, Sensei. 大きいしてください。はい。OK。
Okay. First of all, I would like to introduce my Department of Engineering. The Faculty of Engineering at the University of Miyazaki is located in the rich nature environment where you can see the ocean and the mountains from the building. One of the major projects of the engineering department is the development of solar power generation and hydrogen fuel cells, as shown here. As shown in this slide, Faculty of Engineering has 6 courses in undergraduate, 3 courses in master course, and 3 departments in doctoral course. The Faculty of Engineering consists of the faculty 14 professors, 34 associate professors, and 15 assistant professors, for a total of 19 faculty members. The number of students is as follows: undergraduate 1,564, a master course student 282, a doctoral course student 92 students. So my faculty is the largest number of students in University of Miyazaki. I am a member of the Chemical Engineering Laboratory in the Department of Applied Chemistry. My laboratory members, Professor Oshima and Dr. Inada, and me. And Professor Oshima and Dr. Inada's research team is mainly in the field of DDS, drug delivery systems. And I am in the field of the separation engineering, such as adsorption and solvent extraction. Currently, I am working with 30 students and enjoy our research. So I will begin today's presentation. The contents of the presentation are shown here. First, I will talk the environmental pollution in Japan. About 50 years ago, there are many environmental pollutions in Japan. There are Minamata disease in Kumamoto and Niigata, Itai disease in Toyama, and Yokkaichi asthma in Mie. Except for Yokkaichi asthma, its environmental pollutions of river and sea water by metal contamination. Minamata disease is a very famous Japanese environmental pollution in the world. 20th mid-century, Minamata disease was occurred by the chemical company. Japanese famous chemical company Chisso and Showa Denko was used to synthesize from acetylene to acetaldehyde using inorganic mercury as catalyst. The chemical reaction was shown here. Wastewater, including methyl mercury produced as byproduct, was wasted in the sea without water treatment. Methyl mercury discharged to the sea was bioaccumulated by food chain. Finally, people ate mercury high concentrated fishes, and then people were patient with mercury poisoning. Mercury poisoning caused a neurological syndrome. In last year, there is one of the movie very interesting content. The title is the movie is Minamata. Starring is Johnny Depp. The story of the photographer William Urchin Smith, who took a lot of Minamata disease patients in Kumamoto. Have you watched the movie? Next slide shows the Itai disease. Cadmium pollution was discovered in Jinzu River in Toyama Prefecture in Japan, 1955. Itai disease was caused by cadmium poisoning due to mining. The cadmium accumulated in human body by eating contaminated rice and drinking water. The number 5, the Japanese environmental pollution is Toroku arsenic pollution. The pollution occurred at Takachiho town in Miyazaki, my living prefecture. There are arsenic mine in Toroku. Arsenic trioxide was widely mass produced because arsenic trioxide was used as pesticides, medicine, and paint. Large consumption of arsenic causes acute arsenic poisoning leading to death. While on the contrary, long exposure to even small amounts of arsenic produces symptoms of chronic arsenic poisoning and various health problems. Most typical symptoms appear on the skin, such as the photos, the keratosis, and pigmentation. And in Toroku, serious patients suffer from arsenic poisoning. Many progress to bone disease, skin cancer, lung cancer, and kidney cancer. Many patients died for around 50 years. Miyazaki Prefecture Government and Faculty of Medicine, University of Miyazaki, did the Toroku Direct Wellness Observation and examination over 40 years. New chronic arsenic poisoning patients may be recognized through these wellness checkup. Takachiho Town has been restoring the Toroku mine site and improving the rice field soils. The treatment to restore and to improve the rice field soils contaminated arsenic were shown here. Now there are rich water, clean soil, clean air, and beautiful nature in Toroku. So livestock farmers in Toroku are able to work to breeding cattle and growing crops. Therefore, Toroku was restoring the environment and reviving. When we look at the world's environmental problem, India, Bangladesh, Myanmar, and Vietnam were higher value than arsenic contamination of WHO guideline 10 ppb. Long-term uptake of the contaminated groundwater has caused chronic poisoning, keratosis, and cancer of skin, kidney, lung, and bladder. This slide shows the photo of arsenic poisoning patient. Most typical symptoms appear the skin, such as keratosis and pigmentation, like patient in Toroku. These strange symptoms appeared after drinking tube well water. Therefore, arsenic removal from groundwater must solve immediately to supply safe water. Arsenic is semi-metal contaminated in a lot of minerals such as arsenopyrite. There are many places, river and groundwater contaminated of arsenic. Arsenic is well known as one of the elements that highly toxic to human body. So WHO guideline of arsenic for drinking water is less than 0.01 milligram per liter. Arsenic in aqueous solution has two oxidation conditions, arsenic 3, arsenic 5. In aqueous solution, each chemical form changes with pH as shown here. Figure 1 and 2 show the molar fraction of chemical species of arsenic 3, arsenic 5 versus pH. Predominantly chemical species of arsenic 3 is shown a blue line, and non-ionic arsenic form, and arsenic 5 chemical species, predominantly chemical species is two chemical form, pink line and yellow line, monoanion and dianion. We have focused on magnetic materials as adsorbents. They make it easy to separate the adsorbent from the solution after adsorption test, like this. We have synthesized a new adsorbent of adding yttrium, which has a high basicity to a magnetite base. The adsorbent is expected to have higher adsorption performance for arsenic than magnetite. Therefore, our goal is improvement of adsorption ability for arsenic removal and study for arsenic adsorption mechanism. This slide shows the preparation procedure of new adsorbent. The adsorbents were co-precipitation method as shown here. This figure shows the result of XRD analysis on YMAG-X. The main diffraction peak in these XRD patterns were attributed the magnetite. So YMAG is magnetic materials. Specific surface area and the point of zero charge PCC of YMAG-X was shown in this figure. The surface area and the point of zero charge of YMAG-X increased with increasing X. So content of Y is higher than iron is very good materials. Adsorption test and analysis of PZC were carried out by batch method in these slides. This slide shows the adsorption isotherm of arsenic 3 and arsenic 5 using YMAG X at 303 Kelvin. So you can see the all adsorption isotherm of arsenic 3 and arsenic 5 on YMAG X fit well the Langmuir adsorption model. So YMAG 2 red point materials is very good adsorbent for arsenic 3 and arsenic 5 removal. And then the adsorption capacity Qmax and adsorption equilibrium constant K were calculated by the slope and intercept of the linear equation obtained from Langmuir plot. Qmax of arsenic 3 and arsenic 5 increasing with increasing Y per ion, yttrium per ion ratio. So YMAG2 has higher Qmax than the other adsorbent. The literature value of Qmax of iron and yttrium-based adsorbents are summarized in this table. The results of Qmax by YMAG2 in this study are higher than those of value previous reported. So in the following section, we will talk about this study of the adsorption mechanisms. Adsorption site of metal ions as ion exchanger are explained by the slide. In aqueous solution, water molecules are adsorbed in the surface of metal oxide. And then the proton dissociated from the adsorbed water molecule, producing hydroxide group. The hydroxide group are the adsorption site, very important site. The charge of the hydroxyl group on the surface charge, depending on the pH of the aqueous solution. At acidic pH, the charge is positive like this. And at the basic pH, the surface charge is negative charge as shown here. The neutral charge surface state is called of the point of zero charge. Abbreviation PZC. The PZC is a very important factor on the study of adsorption mechanism of arsenic. There are two mechanisms of adsorption of anions by metal oxide. outer sphere complex and inner sphere complex. Outer sphere complex by electrostatic interactions and outer sphere complex formation indicated no shift, the PZC, and the decrease of adsorption with increasing ionic strength. In case of inner sphere complex formation, the PZC of adsorbent move after adsorption, and the adsorption percentage is not affected by ionic strength. This slide shows the result of PZC analysis. After adsorption of arsenic, PZC of YMAG-X moved to low pH. From this result, arsenic 3, arsenic 5 were adsorbed of YMAG-X by inner sphere complex formation of ligand exchange. This figure shows effect of ionic strength on adsorption of arsenic 3 and arsenic 5 with YMAG-2. Adsorption of arsenic 3 and arsenic 5 is not affected by the concentration of sodium chloride. Arsenic 3 and arsenic 5 were adsorbed by YMAG-X by inner sphere complex formation of ligand exchange. This figure shows effect of ionic strength on adsorption of arsenic 3 and arsenic 5 with YMAG-2. Adsorption of arsenic 3 and arsenic 5 is not affected by the concentration of sodium chloride. I'm sorry. This slide shows the result of XPS analysis before after adsorption of arsenic. Arsenic 3 adsorbed oxidized, while arsenic 5 was adsorbed without changing the balance. 48.4% of arsenic 3 adsorbed was oxidized to arsenic 5, and iron 3 was reduced to iron 2. This slide shows the conclusions. Thank you for attention. So I mistake the presentation time I finished my presentation. Thank you very much.
Thank you, ma'am, for making us aware about pollution in Japan and its various hazardous effects. Your wonderful insights on development of iron and yttrium as an adsorbent for removal of arsenic and other hazardous chemicals from aqueous solutions was truly enlightening. The session is now open for discussions.
At the venue, Vasundhara, whether there is any question?
Okay.
Sorry, ma'am. It is for the volunteer at the session. I mean, venue there. Vasundhara?
Yes, sir.
Any queries from the audience there? They can come and they can ask the question.
No queries from our side, sir.
Okay.
Atira?
Yes, sir.
Yeah, you can go ahead, please.
Okay.
Thank you. First Atira, please thank Ore, and she will leave the room.
Yes, sir. Thank you, ma'am, once again for your enlightening presentation on development of iron and yttrium as an adsorbent for removal of arsenic and other hazardous chemicals from aqueous solutions. It was truly a pleasure to listen to you. Thank you once again.
Thank you very much.
Thank you. Thank you.
Thank you, ma'am.
The next session is on the topic Ways to Study Cellular Inflammation by Dr. Betasiwi Purbasari, Faculty of Medicine, Brawijaya University, Indonesia.
Just a minute, Beta Savi. She will, Atira may introduce her or not required?
Yes, sir. We will introduce her.
Yeah, please, please.
I request Rashmita to introduce our guest speaker. Over to you, Rashmita.
Dr. Beta is a neurologist, earned her MD from Brawijaya University, Indonesia, and PhD from University of Miyazaki, Japan in the year 2022. Her recent research focuses on the role of cholinergic agonist in macrophage inflammation modulation. She intends to be a physician scientist that can integrate biomolecular research and medical science to be used in the clinical application for patient care, especially in neurology field. She is currently attached with Brawijaya University Faculty of Medicine as faculty. Over to you, ma'am.
Good afternoon, dear respective professors, honor speakers, and all the participants. Thank you for inviting me to this program and giving me this valuable opportunity to talk about ways to study cellular inflammation with focus on NLRP3 inflammasome. Inflammation is a well-known term defined as an innate immune response to potentially harmful stimuli such as pathogens, injury, and metabolic stress. Inflammation represents a mechanism of body defense toward multiple causes of agent. Based on the origin of the agent that triggered the inflammatory process, it could be induced by the external agent or endogenous abnormal responses. Based on the duration, the inflammation could be classified as acute and chronic inflammation. Inflammation is an essential immune response that enables survival and homeostasis maintenance. As we see here in the acute inflammation process, this acute inflammation process is a healthy response to several danger signals such as these allergic reactions, some chemical irritants, infections, and trauma such as burns, laceration, and wounds or frostbite. In this acute inflammation process, this amplification loop is important and required to generate sufficient responses. Once the stimuli that trigger this response already resolve, it will lead to resolution phase that will end to maintain the cellular and tissue homeostasis. However, in some pathological condition, this acute inflammation process become excessive, as we see here in this chronic inflammatory process. This excessive inflammation triggered this vicious cycle that could be detrimental, and it failed to be resolved, and it keep continue going and lead to this chronic inflammatory disease, such as cardiovascular disease, neurological disease, autoimmune, and some other disease that familiar that related to the chronic inflammatory disorder. This is a schematic representation of inflammatory responses. On these inflammatory responses, there are four main components of the inflammatory process. These are the inducers that trigger the inflammations, and then the sensors that detect the external stimuli, and then the mediators here, and the effector cells. So in this process, the external signals known as the inducers, for example, like bacteria from the infection or injury from the wound or any other stimuli, it triggers the sensors as we see here in the surface of the immune cells. And then the activation of its sensors will trigger the release and formation of some cytokines and also mediators. The cytokines and mediators will recruit and activate other immune cells to come to the damaged area and activate them as effector cells. These effector cells can enhance or amplify this immune reaction, or also can conjunct to next step of immune function, such as adaptive immunity. In physiological conditions, after the external stimuli that trigger this inflammation process already resolved, it will go to the resolution phase and end this process. But in some pathological condition, this inflammatory process keep continue and go into the chronic inflammatory process. Here, as we can see, one of the important effector cells that has important role in inflammatory response is the macrophage. Macrophage is a very versatile cells that can change from M1 or M2 macrophage. This M1 macrophage has important role in pro-inflammatory action of the cells, such as cytotoxicity, release of pro-inflammatory mediator, and also has important role in the beginning of tissue damage. On the other hand, M2 macrophage has tendency to be anti-inflammatory that has important role in immune suppression, immune regulation, and also tissue repair. Here we can see that one of the key players in the M1 macrophage polarization is the NLRP3. This NLRP3 inflammasome is now becoming one of the concern of several studies. Recent studies revealed that this NLRP3 inflammasome has a very important role in high-impact public health diseases that has so many implications in the human organ, such as intestine, joint, brain, metabolism-related disorder, heart, and also liver. Here, once again, we see that monocyte or macrophages also has important role in this NLRP3 mediated inflammasome disorder. NLRP3 inflammasome is part of innate immune system. It functions as a sensor that manage and modulate inflammation as a response to various stimuli. As a consequence, this NLRP3 inflammasome has a very important role in the initiation phase of inflammatory process. So NLRP3 inflammasome is part of the NLR family that is a member of the big family of inflammasomes. One of the members of this NLR family that has a very high implicated in a clinically disease is this NLRP3 inflammasome. Because of that, now NLRP3 inflammasome is regarded as one of the most potential and promising subject to understand more about inflammatory related disorder. NLRP3 inflammasome is a complex. Upon its formation, this NLRP3 monomer will interact with its adapter protein, DAC, through their purine domain interaction. Here we can see. Then, it will recruit its inflammatory caspase 1 through their CAR domain interaction. This structure then will go further into oligomerization process and form this NLRP3 inflammasome complex that can activate further inflammatory cascade process. To produce a functional NLRP3 inflammasome, usually two important steps are required, the priming and activation step. In this priming step, the activation of the receptor will induce the activation of NF-kappa B pathway that will upregulate the pro-IL-1 beta and pro-IL-18. This priming step alone is not sufficient to produce a functional NLRP3 inflammasome. It required the activation step. Any external stimuli that trigger this receptor will induce the formation of the NLRP3 AAC procaspase 1 complex. After the assembly of this complex, it will induce the autocleavage of caspase 1, and this active caspase 1 will induce the cleavage of pro-IL-1 beta and pro-IL-18 into their active form, and this active form will be secreted from the cells. These all of components of NLRP3 inflammasome activation are investigated by our department in our recent studies. So, recent studies from our department revealed that retinoic acid has important role in inhibiting NLRP3 inflammasome induction by acetylcholine agonist carbachol. The foundation of this study is that acetylcholine can modify immune response in macrophage. Other studies also reveal that several acetylcholine receptor agonists, including carbachol, can induce NF-kappa B pathway. This NF-kappa B is a key pathway in NLRP3 inflammasome activation. And our previous study also revealed that retinoic acid is a potent anti-inflammatory substance. So the objective from our study is we want to examine the role of this carbachol as acetylcholine receptor agonist in modulating the NLRP3 inflammasome and also the involved pathway in the macrophage. We also want to determine if retinoic acid is effective in modifying this response. These are the experiments that were conducted to investigate the priming step and activation step of NLRP3 inflammasome. In this priming step investigation, we conducted these 4 main studies. By employing J774A1 murine macrophage cells, we conducted inflammasome and pathway study, NF-kappa B inhibition, acetylcholine receptor inhibition, and retinoic acid co-treatment study. We start our study by treating the cells with carbachol as acetylcholine receptor agonist for 24 hours. So after we seeded the cell, we treated them with 2 different doses of carbachol, and then we extract the cell to get the protein lysate, and we analyze them by Western blot to see the expression of NLRP3 inflammasome protein and also NF-kappa B family. In this Western blot result, we can see here clearly that the expression of the cell that are exposed by carbachol 1 and 10 micromolar, as shown in the second and third column, show increased expression of NLRP3 inflammasome compared to the untreated group, as shown in this first column. This expression of NLRP3 inflammasome are quantified, and we can see here that the increase are significant, almost 2 timefold changes to the control group. Then we want to further investigate the effect of this carbachol to the expression of NF-kappa B protein. As I mentioned earlier, on NLRP3 inflammasome priming step, NF-kappa B is a key pathway on its activation. Upon its activation, here, when the NF-kappa B pathway is activated, the IKB alpha will get phosphorylated and then it will be degraded and release this P65. And then after this P65 phosphorylated, it will go into the nucleus. So here we can see that the cells that are treated with carbachol, 1 and 10 micromolar, show increased expression of phosphorylated P65 and phosphorylated IKB alpha compared to the control group. To confirm our result, we continue our NF-kappa B study by analyze the nuclear and cytoplasmic lysate. As I mentioned earlier, during its activation, P65 will move or translocate from cytoplasm into the nucleus. Therefore, we analyze its own lysate from the cytoplasm and nucleus. We also conduct this P65 immunofluorescence study to see the visual translocation of P65. As we can see here, the expression of P65 in the cytoplasm lysate of cells that are treated with carbachol 1 and 10 micromolar show less expression of P65 compared to control. In the other hand, the expression of P65 in nucleus lysate are higher in the treated group compared to untreated group. So it means that the P65 moved from cytoplasm into the nucleus. That's why here we see higher expression in the nucleus lysate and less expression in the cytoplasm. This immunofluorescence study confirmed our previous finding. The P65 are shown in the green color and the nucleus of the cell are shown in the blue colors. As we can see here, that more P65 are translocate move into the nucleus, as I shown here, compared to the control group. We can see the nucleus are clearly seen in the control group, but in the carbachol group, it's like covered by the P65. So from this study, we can see that carbachol increase the expression of NLRP3 inflammasome and also induce the NF-kappa B pathway. To determine if this NF-kappa B pathway is instrumental in carbachol inducing NLRP3 inflammasome, we conduct this NF-kappa B inhibition study by treat the cell with NF-kappa B inhibitor or NFI followed by carbachol. In this Western blot result, we can see that the expression of phosphorylated P65 are inhibited in the presence of NFI, as shown in the second, fourth, and sixth column, confirming the inhibition of NF-kappa B pathway. This inhibition of NF-kappa B pathway resulted in the decreased expression of NLRP3 inflammasome. As we can see here, that the increased expression of NLRP3 inflammasome that are induced by carbachol 1 and 10 micromolar that are shown in the third and fifth column are abolished by the NFI pretreatment as shown in this fourth and sixth column. So from this result, we can confirm that NF-kappa B pathway is instrumental in carbachol-induced NLRP3 inflammasome expression. As you know that carbachol is one of the acetylcholine receptor agonists, and it can work through the nicotinic and muscarinic receptor. Therefore, we conduct this acetylcholine receptor inhibition study to see the role of the receptor that are implicated in this process. We treated the cell with the acetylcholine receptor antagonist followed by carbachol. Because acetylcholine receptor antagonist has a very short half-life, we conducted the study in a shorter period as 1 hour. So first, we want to check whether short-term exposure of carbachol is sufficient to induce the expression of NLRP3 inflammasome. And then we co-treated them with atropine or MMA, the antagonist of muscarinic and nicotinic receptor respectively. From this Western blot result, we can see here that even short-term exposure of carbachol actually is sufficient to induce the expression of NLRP3 inflammasome. And then we continue with the co-treatment of the acetylcholine receptor antagonist to see the effect of the role of the receptor in this process. As we can see here, that the increased expression of NLRP3 inflammasome that are induced by carbachol that are shown in the second column are abrogated by atropine as shown in this third column, but not by MMA. These results show that muscarinic receptor has very important role and in carbachol-induced NLRP3 inflammasome expression. Then we continue our study by see whether retinoic acid has a role in modulating this process by treating the cell with RA or retinoic acid, then followed by carbachol. From this result, we can see that the increased expression of NLRP3 inflammasome that induced by carbachol 1 and 10 micromolar that are shown in the third and fifth column are attenuated by retinoic acid pretreatment as shown in this fourth and sixth column. Then we want to see the effect of the retinoic acid in modulating this effect on NF-kappa B protein expression. As we see here, that the expression of phosphorylated P65 and phosphorylated IKB alpha that are increased in carbachol group are also attenuated by retinoic acid pretreatment. Our immunofluorescence study confirming our previous result. Here we can see that the increased nuclear P65 translocation in carbachol group are inhibited by the presence of retinoic acid. From this result, we can confirm that retinoic acid attenuated carbachol-induced NLRP3 inflammasome expression by inhibiting NF-kappa B pathway. So our summary so far is that we found carbachol increases NLRP3 inflammasome and NF-kappa B protein expression, that this effect is diminished by NFI and atropine, and the addition of RA significantly reduces this carbachol effect. We continue our study by investigating the activation step of NLRP3 inflammasome by conducting NLRP3 AAC K-space1 colocalization, K-space1 activity, ELISA, and also employing human cells. The principle of this colocalization study is, as I mentioned earlier, on its activation of NLRP3 complex assembly, this NLRP3 will interact with AAC and then recruit K-space1. So this formation of NLRP3 AAC K-space1 complex are observed by this colocalization study. After we treating the cell, we stain them with the respective antibodies and counterstain the nuclei with GAPI, and then we see the image by confocal microscope. Here is the result of NLRP3 and AAC colocalization study. The fluorescence of NLRP3 that are shown in this green color and the AAC that are shown in the red color, and also the merge between them that are shown by this yellow color, are the highest in the carbachol group. It is reduced by RA pretreatment and nullified by NFI. The case-based 1 NAC colocalization also saw similar fashion result as we see in the previous study. The immunofluorescence of case-based 1 that are shown in this green color and ASC and also merged between them are the highest in the carbachol group. It is reduced by RA and completely abolished by NFI. So from this result, we can see that retinoic acid could attenuate the carbachol induce of NLRP3, ASC, and case-based 1 colocalization formation. As we know that this NLRP3 ASC pro-case-based 1 complex that active can induce the autocleavage of case-based 1. The activity of this case-based 1 are observed by this case-based 1 activity assay. After we treated the cell, we stained them with FLICA. It's a red fluorescent labeled case-based 1 inhibitor reagent to see the case-based 1 activity, and we counterstain the nuclei with host 3342. This is the result of case-based 1 activity assay. The active case-based 1 are shown in this red color. As we can see here that the highest activity of case-based 1 are observed in the carbachol group. It is reduced by RA and completely abolished by NFI. The active case-based 1 will cleave this pro-IL-1 beta and pro-IL-18 into their active form that will be secreted from the cell. Based on this, we conduct ELISA to quantitatively measure the amount of IL-1 beta and IL-18 that are secreted from these treated cells. From this result, we can see that carbachol induce IL-1 beta secretion significantly higher than control group. With increased IL-1 beta, secretion are attenuated by RA and nullified by NFI. IL-18 ELISA result show similar pattern while the NFI group was on par with control group. Finally, we confirm our previous finding by employing these human cells by using THP1-derived macrophage cells. So we differentiate these human THP1 monocyte by treating them with PMA for 24 hours into macrophage. We confirm their differentiation by visually see the attachment of the cell to the flask of culture. Then we conduct the study by pretreating the cell with RA or NFI and also acetylcholine receptor antagonist followed by carbachol. These Western blood data show similar fashion result as we observed in murine macrophage cells. As we can see here that the increased expression of NLRP3 inflammasome that induced by carbachol 1 as shown in the second column are abrogated by RA and NFI as shown in this third and fourth column. Because IL-1 beta and IL-18 are secretory protein, we continue our study with ELISA. In this ELISA result, we can see that carbachol also induced the secretion of IL-1 beta and IL-18. This increased secretion of IL-1 beta and IL-18 as shown in the second bars are abated by retinoic acid and NFI as shown in this third and fourth part. Then we also want to confirm the involvement of muscarinic receptors on this process. These results show that the increased expression of NLRP3 inflammasome that are induced by carbachol, as shown in the second column, are negated by atropine. This result confirmed the role of muscarinic receptor in carbachol-inducing NLRP3 inflammasome expression. Our summaries are that NFI and RA could attenuate carbachol-induced of NLRP3 AAC caspase-1 colocalization, caspase-1 activity, IL-1 beta, and IL-18 secretion. The studies that are investigated on human THP1 monocyte-derived macrophages also show similar results. So our study revealed three important findings. Our first finding is that carbachol enhances the activation of the NLRP3 inflammasome. Recent study revealed that acetylcholine that are synthesized by immune cells has a very important role in the regulation of immune function. Meanwhile, other studies also revealed that cholinergic agonists can enhance the pro-inflammatory response from the cells, such as the phagocytosis and PGE2 released from macrophage, IL-8 released from neutrophil, and TNF-alpha and IL-1 beta released from dendritic cells. Our second finding revealed that NF-kappa B pathway and muscarinic receptor are instrumental in carbachol-induced NLRP3 inflammasome activation. As we know, NF-kappa B pathway is a cellular network that has very important role in inflammasome regulation. Several studies also revealed that this pathway could be modulated by bioactive compounds. Our study confirmed this result, as carbachol is one of the bioactive compounds that can modulate NF-kappa B pathway. Other studies also revealed that muscarinic receptor agonists can activate NF-kappa B pathway through phospholipase D, IKK alpha beta, and also IKB alpha. Our third finding revealed that retinoic acid abrogates the effect of carbachol-induced NLRP3 inflammasome activation. As vitamin A derivative, retinoic acid has many important cellular functions, including immune function regulation. Some other studies already revealed that retinoic acid has anti-inflammatory properties. And earlier studies in our department revealed that retinoic acid negatively regulates pro-inflammatory response in macrophages by inhibiting phosphorylation of IKK alpha beta, IKB alpha, and P65. So our study concluded that acetylcholine can induce the NLRP3 inflammasome activation through NF-kappa B signaling and muscarinic receptors, while retinoic acid antagonizes this effect. So recent studies revealed that many of drugs now are agents that can has capability to inhibit the NLRP3 inflammasome on this pathway. And our study revealed that retinoic acid can also inhibit this NLRP3 inflammasome pathway by inhibiting this NF-kappa B pathway. These are several NLRP3 inhibitors that now is under the trial phase for treating the inflammatory related disorder. Some of them are still in the preclinical trial step. Some of them already go to phase 1, phase 2, even already finished this phase 3 trial step. Targeting inflammation by using nanomedicines offers a promising solution for the treatment of inflammatory disease. These are the nanoparticles that can be used as one of the agents that can modulate or as a strategy to treating the inflammatory disorder through their main therapeutic target. This nanoparticle could be naked like this or functionalized by targeting some moieties through coating group, antibodies, or other affinity ligands. And this will allow them to target passively through the leaking vasculature in inflammatory condition or actively through their main actors of inflammation such as cellular, such as macrophage or endothelial cell, and also circulating proteins. These are also some of nanomedicines that are used to treat several diseases such as neurodegenerative and cardiovascular disease, and then skin or ocular inflammation, inflammatory bowel disease, and some other inflammatory related disease such as arthritis. In the future, the collaboration among clinical medicine and preclinical area and also biotechnology and biomedical field will give a very promising and innovative improvement that could be applied in several aspects such as biological research, clinical diagnosis, and also drug development. Thank you very much for this opportunity. Danyofa dagalu and domo arigato gozaimashita. Thank you.
Thank you, ma'am, for your very informative and enlightening presentation on the role of NLRP3 and NLRP3 complex in the production of IL-1 beta and IL-10, the effect of carbachol on inflammasome protein expression, and P65 nuclear transformation. It was truly a pleasure listening to you. The session is now open for discussion. If the audience has any queries, you may ask now.
Dr. Praveen here. Dr. Bethasvi, very good piece of work. Nice presentation. You said which receptor is that? You mentioned the receptor, muscarinic receptor. Can you hear me? Yeah.
Yeah. Hi. Yeah. Which
receptor is that?
NLRP3 inflammasome?
No, receptor. Cellular receptor.
Cellular receptors? NLRP3 inflammasome. NLRP3 itself actually is a cytoplasmic or cellular receptor.
No, the membrane receptor.
The membrane.
No. My question is, did you check beta integrins there?
In our research, we didn't check that beta integrin receptor. So we are checking those receptor or the protein that are involved in NLRP3 inflammasome activation only. So the inflammatory receptor, it could be bind to the membrane. For example, TLR or some other membrane that are bound to the membrane. But this NLRP3
are you able to link this inflammasome with the specific receptor? So that's what I want to know.
Specific receptor like membrane receptor?
Yes.
In our study, we didn't conduct that linkage study with the external membrane receptor study. But in some other study, they can also see like the connection of this pathway with some membrane receptors such as TLR receptors that so I will...
Okay. And see, these are all, as you have already mentioned, these are secretory proteins. Okay,
Yeah,
so
yeah.
many of these inflammatory cytokines are secretory in nature. Did you try to block this autocrine aspect of these cytokines?
So we have not conducted that study, like to see whether the secretory protein and its like paracrine effect or autocrine effect. We didn't conduct that study. We only see the amount of the secretion of this IL-1 beta and IL-18 by ELISA, but we didn't conduct that study. Like we extract this secretory protein and we treat it to other cells to see the effect as paracrine
or blocking it. Or so with all these, the mechanistic study which you have designed.
Right.
So where you can block this autocrine effect of already flowing these cytokines.
Yeah, that is actually a very good advices to future study of this. So what we give this is that we block the pathway in this NF-kappa-B pathway and also the receptor of acetylcholine, because carbachol is acetylcholine receptor agonist. So the receptor that we study here is the receptor that block the receptor of acetylcholine and this NF-kappa-B pathway. But those receptors, the agent that to block the effect of the secretory protein, we didn't conduct it.
Okay. Thank you. Thank you very much.
Thank you very much for your question.
Is there any other questions from especially from the students, please?
Vasundhara, are there any questions from the students?
Yes, we do have a question. So the question is, what is the best supplement to reduce inflammation?
Thank you for the questions. So the question is, what is the best supplement to reduce the inflammation? So to modify this inflammation process, we can give the strategy to modify the inflammatory response. We can give it as preventive of the inflammation. So, for example, retinoic acid is, many studies revealed that retinoic acid could be used as preventive agent to prevent the further inflammatory process that going to be into chronic inflammation. So it's prevent the acute inflammation to be excessive or detrimental. The other inflammation modulation therapy or strategy, also we can apply them in this pathway. For example, some of the agent now already approved is anakinra and canakinumab, that some of these agents already tried in this COVID-19 treatment several months ago, which this agent can prevent the activity of this active secretion of IL-1 beta and IL-18. So if the question is what kind of agent that used in this inflammatory process, actually it's case by case we see the disease, whether it's still in the early phase or active phase, or maybe already progressed into a severe case. So based on that, we can decide which agent we use, which step we want to prevent or stop. Thank you.
Are there any more questions?
Thank you so much, ma'am, for answering. We have one more question from the student side. Can we use RT-PCR to assess inflammasome inhibitory activity?
So yeah, the answer is yes, we can. So this is actually is our PCR result to see the IL-1 beta and IL-18 expression in the DNA RNA level. So we did this PCR, but this data is not shown, published in our paper. It's submitted as supplementary data. As we see here, even from PCR result in RNA level, we can see that carbachol also induce the expression of IL-1 beta IL-18, and it is inhibited by NFI. So the answer is yes, we can do that. Thank you.
Thank you so much, ma'am. No more queries from my side.
Thank you.
Thank you, ma'am, once again for your enlightening presentation. Up next is a talk on the identification and characterization of biomarkers and therapeutic target for triple-negative breast cancer by Dr. Praveen Kumar Shetty, Director, R&D, Nitte Team to be University. I call upon Ms. Pratiksha to briefly introduce Dr. Praveen Kumar Shetty. Over to you, Pratiksha.
Praveen Kumar Shetty is currently working as Director, Research Nitte, deemed to be University, and Professor of Biochemistry in K.S. Hegde Medical Academy under Nitte. Dr. Shetty has 25 years of experience in teaching medical biochemistry to PhD, medical, dental, and physiotherapy graduate and postgraduate students in India and USA. He was the coordinator, BICEP Department of ITBT Government of Karnataka and visiting professor of UNT Health Science Center at Fort Worth, Texas, USA. He has completed his postdoctoral studies in University of Texas Health Science Center at Tyler, Texas, USA, and University North Texas Health Science Center at Fort Worth, Texas, USA. His postdoctoral research activities mainly include molecular mechanism of fibrinolytic components in lung cancer and other lung disorders, purification and characterization of mRNA binding proteins. His group has identified the novel role of P53 as a mRNA binding protein and its role in regulation of UPA, UPAR, and PI1 and studied the regulation of fibrinolytic components in different animal models like lung injury, infection, and cancer. During his stint in UNT HSC, studied the expression analysis of various biomarkers in breast cancer and prostate cancer. After coming back to India, he has established collaboration with many research centers in India and abroad. Currently, he is working on multiple projects like triple-negative breast cancer, prostate cancer biomarkers, limbal stem cells, ex vivo growth and characterization, rheumatoid arthritis, and Alzheimer's disease. Dr. Shetty's medical biochemistry expertise, his overseas exposure and association with various clinicians has made his try some translational work like ex vivo growth, characterization, transplantation of limbal stem cells, melanocytes in vitiligo patients, chondrocytes, extraction and characterization, synovial fibroblast extraction, growth and delineation of molecular mechanism in rheumatoid arthritis, Wharton jelly stem cell extraction, growth and characterization, screening of novel organic molecules, PLGA nanoparticle formulation and characterization, and metabolic disorder screening and further workup. He has completed many extramural research projects from Cancer Research Foundation of North Texas, ICMR, DST-SERB, VGST, DBT, Rheumatoid Arthritis Research Foundation, and Rajiv Gandhi University Health Science Research Funding. Dr. Shetty has more than 60 peer-reviewed publications in high-impact factor journals like American Journal of Respiratory Critical Care Medicine, Molecular Cancer, British Journal of Cancer, Scientific Report, Journal of Biological Science, American Journal of Respiratory Cell and Molecular Biology, Neoplasia, Journal of Pathology, etc. Over to you, sir.
Thank you. Thanks for kind words. A long introduction. Thank you. Yeah, I'll just share my slides. Can you see my slide? Hello? Hello?
Yes, sir, it's visible.
Okay. Yeah. Can you see my slide? It's full screen, right?
Yes, sir, it's visible.
It's full screen, right?
Yes, sir.
Full screen, right?
Yes,
No,
sir.
no. Is it full screen, Atira?
It's full screen here.
No, in system it is not full screen.
Oh, it's not full screen. 1 minute. Because I have here 2 monitors, that is giving little trouble.
Sir, you can click on that book symbol next to the full screen.
Yeah, the book symbol.
There is a book symbol next to the percentage slider. Book kind of symbol. It will maximize. Not full screen, but maximize. You can toggle easily.
No, maybe my this thing is a problem. How to remove this?
Sir, right bottom. Hi.
Not at come.
Now it is okay, I think.
Okay, yeah.
No, I don't know.
Is it okay?
Certainly.
It's
No?
visible, sir, but it's not full screen yet.
Praveen sir, right bottom.
No, I
near
have 2 monitors here, so maybe that is giving little trouble.
Sir, okay, sir. What we can do is we can make it easier. Right bottom, next to the comments, there is a book symbol, right?
First one.
Third one.
Okay. Double
There is
click.
an open book symbol. Open book, a book open kind of symbol.
Yeah,
next to the notes comments.
It is not working.
I think you have not shared full screen, sir. Maybe you have done full screen, but you have shared.
No. It is in another monitor. It is showing full screen.
Next one, sir. You clicked the first option.
1 minute, I'll come back. I have to take out that. Should go there. Same problem.
Praveen sir, come back to the paper.
Can you see the slide now?
Yes, sir. It's perfectly all right, sir, and audible also, sir. Please.
Yeah, no, but it is not moving.
You click on you click on the screen, sir. You click on the screen, sir.
Okay. Yeah, it's moving, right? Yeah.
Yes, sir.
Yeah. I'm sorry. I'm sorry. Thank you. Thank you for that nice introduction. At the outset, I would like to thank SSP, Dr. Harish Madhyastha, and all the faculty from Miyazaki University, and also our own NMAMIT, the principal, Dr. Chiplunkar, and our biotechnology department, all the faculty members from biotechnology department having me here, giving me this opportunity to share the data from one of our project, triple negative breast cancer. When I was discussing with Dr. Harish Madhyastha, he said, can you take one of these, especially for the students? I said, okay, sir. But this is the research which we have conducted, and most of these data will be there. Is it that okay? Yeah, that's fine. Very good. So we can have this triple negative breast cancer as one of the presentation today. See, so especially for the students, so if they want to, start with a cancer project, the most of the time, the cancer project is now what I am seeing here is this extract, that extract, and then straight away MTTSA. Yes, it is killing the cells. But so we can do a systematic study. What extract, even if you are taking extract, if you are taking molecule. So if you are identifying biomarker, what extract you should know what extract you are taking. So if better you fractionate them, you should know before treating it to the cells or before giving it to the animals, you should know what are the molecules present in it. So if these molecules are present in it, then how it acts, then what model, even if it in a cell line, which cell lines to take. See, most of the studies, if you read, I advise these students, so when you are starting your research, PhD or even after PhD, so read little higher, impact journals. So there, so you will come to know, okay, so this particular model system, so this cell line we have to take. But what we are seeing now is some MCF7 is easily available. So, you put everything on MCF7 and say that the conclusion is, it kills MCF7 cells or it kills cancer cells, and it is an anti-cancer molecule, anti-tumorigenic molecule. But so we can do systematic study. So if you know what extract we are treating, if you know what molecule we are treating, and if you know what cell line model it is, and what is the main oncogene which drives the progression of the cancer in that specific cell line or in that specific cancer. So then, this particular molecule, if it kills the killing is, it's a functional assay. Killing assay is a functional assay at the end you should do actually. So before that particular oncogene, whether it can modulate, whether it can decrease, increase, whether it can decrease the function of that oncogene, whether it can decrease the proliferation rate, whether it can decrease the proliferation, signaling molecules, proliferation, downstream molecules, It can increase the apoptotic molecules. So if you can then correlate, yes, not only this protein level and not only this protein function, at the terminal functional level, this particular molecule kills the cells, means it is apoptotic in nature. It switches off the proliferation. It induces the apoptosis. So by that, the cancer cell death takes place. So this is a complete story. So I advise the students to start if you are taking cancer biology topics, so your approach should be like this. So here we'll be discussing one of the project we started in maybe 10 years. So we are working on that, the triple negative breast cancer progression. So how the triple negative breast, we identified one marker. So that story we'll be discussing today. So this is our Nitte University. Okay, coming to breast cancer. So you have studied this. Breast cancer is the leading cause of cancer death. And so there are different types of breast cancer: the HER2 positive, ER positive, HER2 ER positive, HER2 ER PR positive, and all these things you studied. Because of the rapid advances in the breast cancer field in last 10 to 15 years, we could identify the oncogene molecule or the receptor molecule that is responsible for the progression of the cancer in these different phenotypes. And respectively, the therapeutic molecule identified will act on this particular oncogenic molecule and decreases the activity of this particular molecule. So thereby, the decreases the progression. So thereby, the patients, so which they are taking this molecule, so their survival ability is increasing. Survival ability is increasing. Say for example, HER2. So HER2 positive is one of the leading cancer in India, and also ER positive. So HER2 we have trastuzumab, that is Herceptin. It is called Herceptin. See, easily, even after diagnosis, 20 years, 10, 20 years, easily, they can live, the patients can live. And also tamoxifen, very cheap molecule. It acts on ER positive, anti-aromatase inhibitors. Because of this, the mortality rate has come down in last 10 to 15 years. At the same time, we are seeing some other type of phenotypes, almost 10 years now. These phenotypes, if it is ER positive, we have molecule. If it is PR positive, we have ER PR, we have. If it is HER2 positive, we have molecule. So these phenotypes which are surfacing are negative for these known biomarkers. Negative for these known biomarkers. So we don't have molecule. Of course, we cannot diagnose because we don't have biomarker. So if there is no biomarker, there is no therapy. So especially, so I was associated with one of the hospitals, a cancer hospital in Hubli. There, the director of the Cancer Research Institute, Dr. B.R. Patil, he used to say, see, even for the immunohistochemistry to check HER2, ER, and PR, so that costs about 2,500 to 3,000. That also we are not doing. The patients cannot afford that, and straight away, we are giving tamoxifen that is very, easily available. So if it is ER positive, so their survivability, they are lucky, so their survivability increases. So if it is HER2 positive later on, so if they do immunohistochemistry and find out that it is HER2 positive, so then tamoxifen is the only therapy. But in triple negative breast cancer, how they diagnose, so if the age when they diagnose, if it is less than 50, less than 45, they say 75% is this one will be triple negative breast cancer. So it affects at the early age, and no established biomarkers, no specific regimens or targeted therapies for this triple negative breast cancer except chemotherapy. So initially, it will work, and then there will be resistance, and the patients will die within 3 to 4 years. In that respect, we were thinking, we have to come up with one molecule, and we were working on, see how the research question, you will design the research question, or the research question comes into, if you are focused in that particular area. So this is a good example, actually. this annexin-A2, we were working on this annexin-A2 for long, but not for breast cancer. So with prostate cancer, because metastatic prostate cancer annexin-A2 level is very high. So this is the calcium-dependent phospholipid binding protein expressed in diverse tissues, most of the cell types. We were working on this particular annexin-A2, and we were working on prostate cancer project. So there I wanted one positive control for annexin-A2. Then I opened my freezer, and there I found out that this MCF7 cell lysate for used for some other this thing. So then some HER2 positive SKBR3 cell lysates and all. So I initially started using that as a positive, and annexin-A2 will be there everywhere like that. But to my surprise, the MCF7 didn't show annexin-A2. SKBR3 didn't show annexin-A2. BT didn't show annexin-A2. So what is going on? Maybe my experiment in a Western blot, is the problem, or my assay is the problem. What is going on? So then I used prostate cancer, metastatic prostate cancer cell, PC3 cell lysate, and then we got annexin-A2. Then that question was there. So why it is not there in MCF7? Why it is not there in HER2 positive? So then it is there in the prostate cancer. Then the question was like whether it is expressed in TNBC or not, triple negative breast cancer or not. Before going to that, so this annexin-A2, what exactly the function of this annexin-A2? I forgot to mention that. So this annexin-A2, is a receptor for tissue plasminogen activator. So tissue plasminogen activator acts on plasminogen and converts plasminogen into plasmin at the endothelial cell surface. Okay, so plasminolysis is its function. It's a receptor for TPA. Usually it is synthesized in the cytosol, and it will be transported to the inside of the membrane, and then again it will come out. So that how the exocytosis of this annexin-A2 is taking place. So so our old lab, so we studied that, and that exocytosis, how it comes out. So actually it is not a secretory protein. It doesn't have a signal for the secretory protein function. So but still it comes out to the surface of the cell surface. So there it functions as plasminogen activation, regulation of cell focal adhesion, cell migration, actin cytoskeletal rearrangement, tumor invasion, and metastasis in cancer. And in the cytosol, it does exocytosis and endocytosis. It's transport of immunoglobulin, ion channel activity, and inside the nucleus. So our old lab, they have identified this primary recognition protein complex. To some extent, it also acts as a transcription factor. So till then, so there were many studies on prostate cancers when we started this. There were many studies on prostate cancer. Only one study, Dr. Sharma from Philadelphia, he reported that this particular function of TPA and annexin-A2 is helpful for the cancer invasion. Only one study. So that too, he said it is in other types of cancer. So we asked this question whether this annexin-A2, it is not there in MCF7 when we found out initially, when it is not there in SKBR, the HER2 positive cells, whether it is present in TNBC or not. For that, what we did is we collected some cells, some HER2 positive, some ER positive, some triple negative, many triple negative breast cancer cells. So our initial question was, so annexin-A2, it is present in the prostate cancer metastatic cells like PC3, D1, 4, 5 and all, but it is not there in ER positive and HER2 positive cells. Whether it is right, or what is the expression status in triple negative breast cancer. So this particular panel says that MCF7 ER positive, null or low expression, BT47 cell 4 is a HER2 positive cell, null or low expression, SKBR3 HER2 positive cell, null or low expression, HCC1569 null low expression, HCC1187 low expression. So all other, this MB231, HCC38, 1143, BT549, HCC1937 and 70, all these are triple negative cells. Abundant expression of annexin A2 was found. So now it's clear. Triple negative breast cancer, in triple negative breast cancer, abundant expression of annexin A2 is observed, but it is not there in HER2 positive. It is very low expression in HER2 positive, near null expression in ER positive, MCEP7 and HCC1500. So ER positive cells is not there. So in cancer biology, so when the question like this strikes, so not in the cell line or not in the animal, straight away you should go to, so even for any conditions, clinical condition, I suggest, so go to that particular clinical condition and confirm your hypothesis there. Confirm your hypothesis there. So we should go to the clinical. Before going to the clinical, so the question was there. So whether at the RNA level also the same expression was there or not, we checked it. Yes, the RNA level also same expression. And in the cell where it is expressed at the cell surface, cytosol, or it is expressed in cytosol as well as cell surface. We did immunohistochemistry with the cell pellet before going to the clinical samples. Immunohistochemistry with the cell pellet, where you can see here, so where HER2 is near null or low, so annexin A2 absorption is seen in this particular cell. So then in another cell where HER2 expression is very high, annexin A2 is low. Annexin A2 is low in this immunohistochemistry. So then we went to this, we wanted to check the clinical samples. We did many clinical samples, HER2 positive also, ER positive also, annexin, the triple negative breast cancer also. So this representative data shows that the annexin A2 is abundantly expressed in the triple negative breast cancer. And also it increases with the progression of the breast cancer, increases with the progression of the triple negative breast cancer. So now at the clinical level, we are telling that annexin A2 expression is, there is a differential expression of annexin A2 only in triple negative breast cancer. And the expression of this particular biomarker increases with the progression of the triple negative breast cancer. And it is reciprocally regulated. When HER2 is there, annexin A2 is not there. When annexin A2 is there in the specific tissue sample, the HER2 is not there. So these two are reciprocally regulated. So that gives the idea that in HER2 negative, that time we used to call it as HER2 negative breast cancer. So triple negative, there are many classes now, mesenchymal stem cell, basal stem cell like mesenchymal, ER positive, P53 positive. There are different classes of triple negative breast cancer now. But at that time we checked, when HER2 is there, this particular expression is not there. When this particular annexin A2 expression is there, HER2 is not there. So then many clinical samples we could demonstrate that. So these two are reciprocally regulated, and that we published in PLOS One at that time. And in addition to this, in this same paper, same article, okay, annexin A2 normal function is working as a receptor for TPA. In addition to that, some 1991 paper was telling that, one article was telling that, some association of EGFR. They didn't say there is interaction. Some association of EGFR is there. So then our next question was whether this annexin A2 is interacting with EGFR. I'm not putting all the data. Very, very important data from these papers I'm putting. Yes, we could immunoprecipitation and reverse immunoprecipitation. We could show that EGFR antibody could pull annexin A2. Annexin A2 antibody could pull EGFR at the membrane surface. EGFR, most of it is there in the membrane surface. Annexin A2 is there in both membrane as well as cytosol. At the membrane surface, annexin A2 is interacting with the EGFR. And we did site-directed mutagenesis also. We had all these plasmids. So this is a wild type annexin A2, then we transpected Y23F, that is actually, if annexin, that time, this was the hypothesis, proven hypothesis. at the 23 phosphorylation, 23 tyrosine gets phosphorylated and it goes to the membrane surface. Said like that. So what we did is at 23 we put phenylalanine. So side-directed mutagenesis, we put phenylalanine in the place of tyrosine. So we didn't get anything, so with EGFR, so that particular annexin-A2 didn't come. It could not full this negative phenylalanine containing Y23F annexin-A2. So Y23E annexin-A2. So this is called phosphomimicking. So tyrosine, in place of tyrosine, we could put glutamic acid. Glutamic acid has two carboxyl groups. So phosphate, so two negative charges. So even if you put two carboxyl groups containing amino acid, that acts as a phosphorylated protein. So Y23E, this phosphomimic, could come to the membrane surface and also could interact with the EGFR. So this is vector control. So very little wild type annexin-A2 we could see. So that means this specific phosphorylation of annexin-A2 is important. It has to come to the membrane surface, then it will interact with the EGFR. Only that much, or it has anything to do with the EGFR downstream signaling. So it is known that in triple negative breast cancer, abundant EGFR expression is also taken. That is why we designed this experiment. So for the EGFR function, this annexin-A2 interaction has any role. Okay, so what we did here is we downregulated annexin-A2 and induced with EGF. So ligand, if you put EGFR, phosphorylation should take place. So phospho-EGFR 1068 phosphorylate at the membrane surface we could see, but at the inside leaflet that 845 phosphorylation didn't happen. And so also when annexin-A2 is knocked down by siRNA, when annexin-A2 is knocked down, even with EGF, could not phosphorylate the inside leaflet 845 EGFR phosphorylation. So also P38 goes down, ERK goes down, phosphorylation of ERK goes down, phosphostat3 goes down. So for the EGFR downstream signaling, this interaction with annexin-A2 is critical. So till now what we have proved, annexin-A2 is differentially expressed in triple negative breast cancer. It can be a candidate for biomarker because there is a differential expression. So there is a membrane expression, abundant membrane expression, and also that interacts with the EGFR. For the critical downstream function of EGFR, this specific interaction is very, very critical. Then only EGFR phosphorylation. What about cytosolic SARC phosphorylation? So when you knock down annexin-A2, the SARC phosphorylation is also not happening. So this is the molecule, if we can play with this particular molecule, so we can downregulate the proliferation function of this triple negative breast cancer cells, and we can knock down proliferation, or we can downregulate the proliferation. So, and also maybe apoptosis, we'll see that. So then again, at that time, so we did both. So the Herceptin resistant. So when you add a Herceptin, when you add HER2 antibody, that is trastuzumab, usually is for the molecule which is given to the HER2 positive breast cancer. So these patients will come with the resistance later on. So with the cell line model, we added this HER2 antibody and we checked whether by blocking HER2 antibody, annexin-A2 at the membrane surface increases. So if it increases, so there may be an alternative pathway. So that may be the reason for the triple negative, the resistance during Herceptin treatment. So again, so in all these cells, the annexin-A2 membrane, abundant membrane expression was seen. So JMT1, this particular cell is the Herceptin resistant breast cancer cell. Herceptin resistant breast cancer cell is also same as that of the expression is same as that of the MDMB231 or triple negative breast cancer. So endogenous anyway, it is there in the triple negative, but membrane wash also we could see even in JMT, Herceptin resistant annexin-A2 expression is very high. So then again, so EGFR interaction is there in JMT1 also. Then again, when we knock down HER2 in HER2 positive cells, abundant annexin A2 membrane expression was seen. This is total internal reflection microscopy data. So around 200 nanometers, so upper this thing, we can see the expression. So it is at the membrane surface, not at the cytosol. So then some functions, proliferation function with the EGF and knocking down EGFR we checked. So this annexin A2 antibody we tried, okay. This data is from the British Journal of Cancer. We published whether annexin A2 antibody can be used in these patients at the cell line model we studied, can be used to downregulate the annexin A2 function, to downregulate the EGFR function, to downregulate the proliferation, cell proliferation function and migration and other cancer cell functions. So annexin A2 antibody could downregulate the cell proliferation. So this is MDMB231. So this is JMT1 cell. The second panel is JMT1 cell, where the Herceptin resistant also. So when you use antibody, so that works. So then the wound healing study also we did. So there is the coverage of wound with the annexin A2 antibody. Annexin A2 antibody is used. So the wound coverage is not seen. So wound healing is not seen. So that means the proliferation and migration goes down. Proliferation and migration goes down. So then we knocked down EGFR. That time we didn't see. So that means this annexin A2 antibody blocking works via EGFR. Annexin A2 antibody blocks annexin A2 at the membrane surface of TNBC and HER2 resistant cell line, Herceptin resistant cell lines. So thereby annexin A2 EGFR interaction is hindered. So if this interaction is hindered, so downstream signaling goes down. If this downstream signaling goes down and the proliferation protein level goes down, proliferation protein function goes down, proliferation goes down, cellular proliferation, cancer cell proliferation goes down, cancer cell migration goes down. So then the apoptosis rate increases. So thereby the cancer cell death rate increases. So this annexin A2 antibody can be a molecule which can be used in the triple negative breast cancer treatment. So then again, so up in annexin A2 antibody, when we added annexin A2 antibody, so with the EGF, actually ligand binds to the receptor, that is EGFR receptor, then internalization of EGFR is seen. With this confocal microscopy data shows that, so when you add EGF, that ligand EGFR, internalization won't take place in presence of annexin A2 antibody. So that means it blocks the EGFR function itself. So then again, in both the MDMB231 and JMT1, so all the downstream signaling is going down with the annexin A2 antibody, and so also the migration function and the proliferation function. So this is the British Journal of Cancer data, which I have shared with you. So till now, the project, we started with the annexin A2 antibody, the differential expression in cell line we found. Then we confirmed with all the cells, and we could see in the triple negative breast cancer abundant expression of this particular protein. And it is a normally it is a TPA receptor. Once it comes to the membrane surface in cancer cell, more and more annexin A2 comes to the membrane surface. There it acts as an oncogene. I think the students know about oncogene. So normal proto-oncogene. EGFR is also a proto-oncogene. So the normal protein functions as an oncogene here. So in this particular condition, it interacts with the EGFR. This interaction is very critical. When you block this interaction by annexin A2 siRNA or by annexin A2 antibody, EGFR downstream signaling goes down, proliferation protein level goes down, proliferation protein function goes down, proliferation goes down, and it may induce the apoptosis. Apoptosis data was there. I think in this I have not put. So apoptosis increases, apoptosis rate increases. So this can be annexin A2 antibody can be a molecule to try in triple negative breast cancer. So that is about the annexin A2 role and its interaction with EGFR. Meantime, our question was annexin A2 is getting phosphorylated and going to the membrane surface. We came to know that annexin A2 is also a glycosylated protein. only phosphorylation is must for the translocation, membrane translocation, or this glycosylation is also necessary. So then we found one animal lectin or human lectin, galactin 3. So that is a report that is interacting with the EGFR. So okay, galactin 3 is interacting with the EGFR. Galactin 3 is a glycoprotein binding lectin. Annexin A2 is a glycoprotein. Whether there is any interaction, whether there is an association of expression in the triple negative breast cancer, very few cells we checked, and we found that in MD-MB231 abundant expression of annexin A2 as well as galactin 3 was found, and also in tissue level we checked wherever annexin A2 is expressed, galactin 3 is also expressed, and it is membrane surface. It is expressed, and we did many tissues there also where annexin A2 is expressed, high expression is seen, galactin 3 expression is also seen. Then we checked annexin A2 and galactin 3 are interacting. So galactin 3 could pull down annexin A2, galactin 3 antibody could pull down annexin A2 antibody could pull down galactin 3. So then this particular experiment, we did that experiment at that time. Which one was the upstream molecule here? Whether annexin A2 is the upstream molecule, where the galactin 3 is the upstream molecule. So annexin A2 is the upstream molecule. We knocked down annexin A2, we could see that galactin 3 is going down. So that means that, say, if you block annexin A2 function, galactin 3 function also should go down. Or in this particular project, whether we can play with the galactin 3 also. Can we play with the galactin 3? Can we play with the glycosylation of annexin A2? In that, we designed some experiments in that respect. So in this particular experiment, what we did here is, so this one by one, I'm telling, see the students also can think of, so one by one, the questions, they can raise one by one questions, and they can read in that line, and they can come back and, design the experiments. Okay. So the molecular, now the question is, galactin 3 is a lectin. That means it has some amino acid residues in this particular protein. That amino acid residues specific to oligosaccharide of a glycoprotein. Here the annexin A2 is a glycoprotein. It is reported. So whether because annexin A2 is interacting with EGFR, galactin 3 is interacting with EGFR. We found that annexin A2 is interacting. There is a report that galactin 3 is interacting with EGFR. So these two particular molecules are interacting. We found that these two are interacting. So we thought maybe that oligosaccharide chain present in the annexin A2 is responsible for this interaction. We wanted to block that glycosylation at the cytosol itself. So broad spectrum, the glycosylation inhibitor tunicamycin at 10 micromoles we used at different time points. So membrane wash we checked after putting this tunicamycin. Membrane level is going down. Cell lysate is coming up. Means it is not going to the, not translocating to the membrane surface. It is staying there in the cytosol itself. That means when you block the glycosylation of this particular glycoprotein, if that glycosylation is not taking place, so then it is not translocating to the membrane. Not only phosphorylation, glycosylation is also very, very important for the specific receptor protein to get translocated to the membrane surface to act as an oncogene. So then with the different concentration we checked, even the 5 micromole is good enough. So then with the tunicamycin, we checked the interaction with the galactin 3. So whatever it comes to the membrane, that is interacting with the galactin 3. So if it is not coming, it is not interacting with the galactin 3. So this is lectin, what I have said now. So this has the oligosaccharide chain in it. So the annexin A2, this amino acid residues, specific amino acid residues in galactin 3, it is binding. So we put FITC into this. Sorry, I'll come to that later. So okay, now the story is this: annexin A2 is specifically expressed in triple negative breast cancer. Its interaction with the EGFR is very, very critical for the EGFR downstream signaling function and the proliferation in triple negative breast cancer. When you knock down annexin A2, the EGFR downstream signaling functioning getting hampered, and the proliferation and migration is going down. The cellular cancer cell function is going down. When you use annexin A2 antibody also, that could downregulate the cancer cell proliferation. and the progression of cancer in triple negative breast cancer. So then there we found that the phosphorylation is very, very critical. With the phosphorylation, is the glycosylation is critical or not? We found that glycosylation is also very, very critical. So it gets glycosylated and it comes to the membrane surface. There it is interacting with the animal lectin, galactin-3, and in this further forms a complex with EGFR. This association is very, very critical for the downstream signaling. So then we thought whether we can dissociate this galactin-3 annexin A2 interaction. Can we dissociate galactin-3 and annexin A2 association or interaction? So galactin-3 is an animal lectin. Some amino acids in this particular protein has an affinity to bind with oligosaccharide. So like that, plant lectins are there. Plant lectins are there. So these plant lectins are present, abundant expression of plant lectins you see in all these legumes. So one of these legumes, that is Caesar areatinum. So the chickpea, from chickpea, we extracted, purified and extracted one lectin of 30 kilodalton lectin, and that 30 kilodalton lectin also forms a dimer. It also forms a dimer and also trimer also we got it. So it also forms a dimer, specific dimer. And we put FITC into that lectin. FITC by affinity chromatography we purified, and to this purified lectin we added the FITC, and then we checked whether it is interacting with the annexin A2 or not at the membrane surface. Yes, it is interacting at the membrane surface, and also knocked down with the affinity purification, and then with the annexin A2 antibody knockdown. So we could show that the annexin A2 is interacting with the specific interaction of annexin A2 is there with the galactin-3. So this should be lectin actually. This should be tunicamycin. Yeah, tunicamycin. So when you add lectin, so membrane surface galactin-3, which is coming with the annexin A2 is going down. So that means lectin could dissociate this specific lectin purified from the chickpea, 30 kilodalton lectin could dissociate galactin-3 from annexin A2. So immunoprecipitation. So we treated lectin. So then to the cell lysate, so we added annexin A2 antibody. So this annexin A2 antibody could pull the galactin-3. So then we check the galactin-3 expression with the Western blot. So galactin-3 expression is going down. So then with the galactin-3 expression going down, so 845 phosphorylation is going down, EGFR, SARC phosphorylation is going down, STAT3, phospho-ERK, all phosphorylation is going down. So with the tunicamycin and also with the lectin, so all this level is going down. So that means the lectin at the membrane surface acts like galactin and it competes with the galactin. It goes and binds to the annexin A2. So thereby galactin-3 annexin A2 interaction will not be there. This interaction is very, very critical for the EGFR downstream signaling function. When you dissociate this interaction with the plant lectin, which has more affinity towards this annexin A2, then also EGFR downstream signaling going down, and the proliferation is going. See, BCL2, BCLXL is going down. The proliferation protein level is going down. And then we did DNA fragmentation study. So the DNA fragmentation we could see in 24, 36 hours during lectin treatment. So the wound healing, the wound healing is in control. You can see here. So that wound healing was not seen with the tunica as well as lectin. It was more active. Lectin is more active than the broad spectrum inhibitor of glycosylation, and also the proliferation is going down. So this will be published in molecular and cellular biochemistry. This particular story about annexin A2 is a marker in triple negative breast cancer, and this interaction with EGFR story, and the first time we reported this interaction and critical role of EGFR downstream signaling. Then the annexin A2 antibody story we report in British Journal of Cancer. So then the galactin and EGFR interaction, galactin and annexin A2 interaction, then the purification of this particular lectin and its action in dissociating annexin A2, galactin 3, and downstream signaling. So this we reported molecular and cellular biochemistry. So then, we know that this annexin A2, even though it is not a membrane protein, in triple negative breast cancer, so membrane deposition is seen, and this membrane deposition, some annexin A2 is also getting secreted out. So if it secretes out in cancer cells, so that should enter into the systemic circulation, and we should be able to analyze this. So that was our idea. So yes, it is coming to the membrane surface and also getting excreted. It goes out in the conditional media. It goes out in the conditional media, and in cancer tissue, it goes out from the tissue, cancer cell, to the microenvironment, and then to the systemic circulation. So we could analyze in the serum. So this with the annexin A2, we also took IL-6 and HBEGF. So known cytokine which is overexpressed in the cancer, and HBEGF, the ligand, is overexpressed autocrine markers. So with the annexin A2, so we wanted to see this annexin A2 as a biomarker, serum biomarker. With that, we took IL-6 and HBEGF and its expression in triple negative breast cancer. So annexin A2 and HBEGF expression goes well along. Both are increased in triple negative breast cancer, not in other cancer. But in IL-6 is expressed in any inflammatory, any cancer. So the level increases, but it is not a differential expression. So we knocked down annexin A2 and we checked IL-6, HBEGF. At that time, we are not sure. Now we are very much sure that annexin A2 in different model system, we proved that when you knock down annexin A2, this is MCF. This is just expression. MCF, it is not that. IL-6 is there in all the cancer cells. HBEGF also, it is not there. So only two cells, one ER positive we took and one TNBC. So its abundant expression is seen. It's not there, but IL-6 is there. So then we knock down the annexin A2, IL-6 goes down, HBEGF goes down. And when you induce annexin A2 with any ligand like EGF, so annexin A2 comes up in MCF7 cells. Annexin A2 comes up in MCF7 cells. So we have a different project going on in this respect. So annexin A2 comes with EGF, and HBEGF comes with the EGF. IL-6 comes with the EGF. So abundant expression is seen even in MCF cells. So then the level we checked. So in normal it is 280 cc. Now if you go and check the annexin A2 with whatever the ELISA kits available, it will show around 30-40 nanograms per ml. Now this was the at that time, only one Korean company was, producing this ELISA kit. Only company was producing. We paid the hefty amount for each kit. So not that specific. As it evolves, now more specific, and for lower price, you are getting ELISA kits. And these ELISA kits now the normal level itself is 40. Then the differential anyway you can do that. But at that time it was showing 287, and the normal one HER2 positive. See. Just like cell line, so here also it is going down. The HER2 negative or the triple negative, the level abundant expression of annexin A2 is seen. So it's highly significant, and the differential level also you can see. So then HBEGF level, so normal is this, the HER2 positive level increases, and HER2 negative abundant increase is seen. Then IL-6, as I mentioned, the level increases in both. It's not for the differential one, but annexin A2 can be used for the differential diagnosis of the cancer, breast cancer. So you can confirm it. You can confirm it by serum analysis. This we reported in the Annals of Clinical Biochemistry. So to summarize this, so annexin A2 is a normal functioning protein. Function is it acts as the plasminolytic. It helps in plasminolysis. It acts as a receptor, membrane surface receptor for the TPA to come and bind. So TPA breaks TPA and plasminogen to come and bind. TPA acts on plasminogen and it will be converted to plasmin. So this particular function takes place on the surface of annexin A2. So this is a normal function. Actually, after glycosylation and after a phosphor, it comes to the membrane surface. Then it interacts with the EGFR. This interaction is very, very critical. The abundant expression of annexin A2, EGFR, and galactin 3 is seen in triple negative breast cancer. So if annexinator is interacting with the EGFR, all the downstream signaling goes up, STAT signaling, AKT signaling, ERK signaling, PKC signaling, all this goes.
Yes, sir. Thank you.
Are there any more questions?
No more questions.
Sorry, Dr. Venkatesh, I took little, 10 minutes extra, I believe.
No issues, sir. We are within time limit.
Okay, thank you. Thank you. Thank you very much.
Thank you once again, sir, for your thought-provoking presentation. It was a pleasure listening to you.
Thank you. Thank you,
to-
Professor Praveen Kumar Shetty. On behalf of Miyazaki University, I thank you for giving lecture.
Thank you, sir. Thank you.
We have now come to the end of the morning session. I would once again thank all the speakers for bearing with the time difference and taking the time to share your thoughts and insights on some fascinating topics. I hope that everyone attending the seminar was able to glean information from their valuable insights. We will now be dispersing for a short lunch break. The next session will begin at 2 p.m. I request the audience at Shambhavi Hall to please remain seated for a photo session.
Harish, sir, please be online, sir. We'll be projecting on the screen for the photograph. You'll be in the background screen, sir. Please be online. I'll inform you at time, sir, for your.
I will just ask Professor to be switch on the.
Okay, it will take around 4-5 minutes. Around 4-5 minutes it will take to set up. The students to gather there.
Sir, can I leave
I'll
now?
inform you, sir.
Can I leave now?
Please be.
Sir, I'll leave now. Okay. Thank you. Thank you.
Yeah. Thank you, sir.